primarily at public health aspects and the use of whole genome sequencing in investigating outbreaks of infectious disease. And today we're taking a slightly different focus, and we'll do a bit of scene setting first and introductory, and we'll also look then at how this technology can be applied in the food industry. So as Tracy said, we have three excellent speakers lined up for you this morning. I will introduce them first, and then we'll get started. And if you could hold your questions or put your questions in the Q&A box, which is at the bottom of the screen, and we will take all the questions at the end of the three presentations for the panel. So put the questions in the Q&A box, and we will get to them at the end. So just to introduce our three speakers, Dr. Purnika Ranasinghe is a Senior Scientific Officer in the Pathogen Genomics Unit of AFBI, the Agri-Food and Biosciences Institute in Northern Ireland. She's a microbial ecologist and a bioinformatician specialising in genomics, supporting whole genome sequencing-based bacterial identification, tracing, and early-stage disease diagnostics and control in livestock and wildlife. She is interested in exploring existing and novel biosurveillance methods and detection of genetic variations through pangenome analysis. Our second speaker then, following on from Purnika, will be Nick Andrews, who is Head of Food Safety and Quality for Dawn Farm Foods, which is Europe's largest multi-species producer of cooked and fermented meat ingredients for the food service and food manufacturing sectors. Nick heads a team of food safety and science professionals in assuring the safety and integrity of the company's and clients' brands. He's also an adjunct lecturer with UCD and has led the development of a bespoke food safety management program, Food Plus. And our third speaker then is Dr. John Donaghy, who is Global Head of Food Safety for Nestlé. Having been with Nestlé for over 14 years, he's currently responsible for food safety and global operations, leading teams of subject matter experts and managing an internal food safety audit team. He's also a member of the International Commission on Microbiological Specifications for Food. And before joining Nestlé, he also worked at AFBI. John is leading on integrating advanced digital tools into food safety decision-making, leveraging big data, predictive analytics, and advanced diagnostic technologies. So I think you'll agree that we have some serious experts in the room this morning. So I'm sure it's going to be really informative, and I hope that, and I would encourage people to ask questions, because we do hope for a good discussion at the end of the presentations. So with that, I am going to hand over now to Purnika, who's our first speaker this morning.
Thanks, Linda, for the nice introduction. I think everyone can hear me okay and see my slides.
Yep, all good. Thanks, Purnika.
Yeah, thank you. Good morning, everyone. Thanks for joining the webinar this morning. So I will start with the foodborne illnesses, looking at the status in the UK and the Ireland region in general. So according to the 2018 estimates, nearly 2.4 million cases has been identified as associated with foodborne illnesses in the UK, which is costing nearly £9 billion to the UK economy. So when you look at this £9 million statistics, nearly 2/3 of it is actually associated with unattributed foodborne diseases, which means these foodborne diseases, the factor or the cause has not been clearly identified, which is quite large in terms of identified foodborne illnesses. So this actually highlights how crucial it is to detect, monitor, and characterize these foodborne pathogens in the food production lines and also in the final product to reduce the impact on the public health and also the UK economy. So how do we actually detect and characterize these foodborne pathogens before they spread? So whole genome sequencing is being increasingly used as a primary and a kind of a refined and a mature tool for epidemiological surveillance studies, to study outbreak investigations, and for environmental monitoring in food and food processing plants. Generally, whole genome data are complex, but when you combine it with bioinformatics, it provides the highest resolution for many genomic analysis. So this helps to distinguish pathogens according to their specific strains and identify their antimicrobial resistance traits, and also help to track the sources of contamination very quickly, that is to prevent any regional or cross-border contaminations or outbreaks. And together, all this information actually feed into the global surveillance programs that actually helps to improve public health policies and apply stricter food safety controls to protect public health. So early this year, European Commission released a legislation on foodborne outbreaks, where now EU states, the member states have to use whole genome sequencing to identify foodborne pathogen related to any foodborne incidents or suspected to be associated with a foodborne outbreak. So they came with a long list of pathogens, including Salmonella, Listeria, E. coli, Campylobacter, etc., sourced from food, animal feed, or any related environmental samples. So these kind of legislations are actually uh, improved legislations are actually positively impact to protect the public health, because these are come with clear direction. But at the same time, it's raised the question, how do we make whole genome sequencing accessible for facilities or institutes which they don't have it? So in today's talk, I will focus on a case study that we completed recently on Listeria monocytogenes, to looking at their prevalence, the genetic diversity, and the phylogenetics association within the UK and Ireland. So here I will try to demonstrate how we actually use whole genome sequencing to detect and characterize this particular foodborne pathogen. And we try to emphasize the importance of whole genome sequencing analysis and how we can improve these results in order to investigate foodborne outbreaks at national and international level. So why Listeria monocytogenes? I don't think I really need to go through the slide because the figure itself speaks. Looking at the number, we can say like it's quite large compared to any other foodborne illnesses. In generally, Listeria records are really low. According to the 2018 estimates, it's only 162 cases were reported, but with 26 fatalities. So because of this reason, it holds the highest cost, around 230,000 pounds in general, because of this high number of fatalities. So Listeria monocytogenes is a gram-positive bacteria, and it causes listeriosis, which is a severe foodborne disease in human and animals. If you look at the classification of Listeria, it actually have several lineages. Nearly there are 4 lineages been identified for Listeria, lineage 1 to 4, and within these lineages, they've been further characterized down to different serotypes, sequence types, and their corresponding clonal complexes. So in general, any infection can cause by consuming contaminated food. And generally, any infection can be asymptomatic or appear with mild gastroenteric symptoms in a healthy individual. So, but under certain circumstances, it can develop into a more invasive form which can damage central nerve system, where we identified it as neurolisteriosis. So in general, as I mentioned before, listeria is less common compared to other foodborne pathogens, but it has a massive impact on public health because of high mortality rate, which is nearly 30%. And it also causes neurological damage. So here we have three data sets being collected from Northern Ireland and England. So in Northern Ireland, we have two data sets that's sequenced and analyzed in AFBI. So the first data set is actually include 42 listeria isolate that we actually analyzed and reported in PathSafe project, and 96 other listeria isolated been collected during 2015 and 2016 by our colleagues in Food Micro Unit in AFBI. So the second dataset has been already published by Madden et al. In 2018. So in Ireland, for Ireland, we collected nearly 64 listeria isolate from the public domain, and we try to include all the isolates reported in Hurley et al. In their publication in 2019. So for England, we try to include all the isolates sourced from food, which is included in the LIXIG project. These samples been collected between 2002 and 2014 period. So in total, we included nearly 1,005 isolates covering all three territories, and we analyzed all the isolates through our in-house bioinformatics pipeline Raven Path workflow. So Raven Path workflow is a full-scale data analysis process, which actually include data quality control, genome assemblies, variant calling, sequence typing, serotyping, and phylogenetic analysis. We actually carry out external quality assurance exercises to validate our processes. So I would like to welcome any participants from the audience if they are wish to carry out any EQA analysis to standardize their bioinformatics workflow. Get in touch with us. We are happy to help, and we can do this sample swapping, and we can get our workflows validated in that way. So when you look at the results, so we first took all the 1,005 isolate, ran through our Raven Path pipeline. So after data sanitization, we ended up with 962 isolates covering all three territories. So the first thing we did was to identify how many different serotypes we can determine in this data set. So we used a 5-gene panel in silico serotyping method to determine whether these isolates belong to any particular serotype. So here we identified 4 different serotypes in all three regions, which are actually affiliated to lineage 1 and lineage 2. So as you can see here, the lineage 2 is actually corresponding to nearly 80% of the cases reported across all three regions, which belong to serotype 2A and 2C. So apart from the high prevalence of lineage 2, serotype 2A, there were a couple of serotype 1, lineage 1 serotypes been identified in Northern Ireland and England, which is 4B, which is this one, and also 2B serotype in Ireland. So the second level of classification, we try to look at the multi-locus sequence typing method. So these are all in silico methods. We did not carry out any lab testing or PCR method to determine the sequence types of these isolates. So here we try to determine the pathogen sequence type and their corresponding clonal complexes using a 7 MLST scheme consists of 7 genes. So in total, we identify 41 distinct clonal complexes, including 4 singletons. So the singletons are correspond to only sequence type. So there is no clonal complex information being identified for these. So the most prevalent clonal complexes were clonal complex 121 and clonal complex 9. So if you look at these 41 different clonal complexes, we identified 8 clonal complexes that are common to all 3 regions. And when you look at the higher, the prevalence of different clonal complex in each region, we can see like 39 different clonal complexes been identified in England and 18 in Northern Ireland and 11 in Ireland. So we can see like this is actually corresponding to the number of samples we have included in this study. So more samples actually show more diversity of clonal complexes. So we further analyze these assemblies and isolates through whole genome MLST analysis. So here we try to define the population structure of Listeria isolates using like a predefined or customized core genome scheme, which is actually a set of loci with a single representative allele. And we try to map this information out using minimum spanning tree, which based on MLST, core genome MLST allele distances. So here we try to, so how do we actually read this minimum spanning tree? So each dot represents a single sample, and the distance between 2 dots actually correspond to the allelic differences. So shorter the distance, more closely related they are. So again, when we mapped this information against the lineage, the origin and the serotype and the clonal complexes, we can clearly see how nicely these isolates get clustered to each other. And especially clusters like these, they are closely clustered together. But when you look at the origin, they're coming from 3 different locations, which actually interpret the prevalence of the same clone complex or the sequence type in different regions. Then we try to map this information against the sequence type, clonal complex, and the serotype. So again, like I mentioned before, it is very easily to visualize how these clonal complexes are clustered together, showing the phylogenetic association among these clusters. And although this core genome MLSD is less resolved compared to the SNP approach, we have identified a couple of interesting facts looking at these minimum spanning trees. So one is like, if you look at the clonal complex 9, we actually identify 2 different serotypes identification. So we would like to investigate it a little bit more just to see whether this is something to do with the sequence error or is it actually a true sequence type, serotype that has been identified in this cluster. And the other thing is clonal complex 115, which is actually reported between 2017 and 2023 in Europe, kind of hyped in human cases. This is something that was not clearly reported in this region before. So it was kind of an interesting discovery that we have identified this particular clonal complex in Northern Ireland samples and in England samples. And the other thing, because these isolates get clustered together, depend on their allele differences, it was quite easy to identify any isolate that has not been characterized before. So we were able to match it to its closest relative. So the fourth level of, or the final level of classification, we use the SNP-based phylogeny, that is to characterize the strain based on the SNP distance. So the SNP distance or the single nucleotide polymorphism method is kind of robust against recombination because these bacteria, especially Listeria, Salmonella, they undergo recombination as an adaptation process. So we use the number of SNPs detected in each lineage or each strain as a proxy to determine the genetic closeness of this isolate. So if you look at the branches, the first two branches represent the lineage 1 and lineage 2, and each subcluster is corresponding to clonal complex or the sequence type. And at the tip level, you can determine the genetic differences between each isolate. So as we can see, like we have started with serotyping and sequence typing, and then the core genome MLST analysis, and finally SNP-based analysis to characterize these pathogens, just to get to look at their diversity and their prevalence. And then we further analyze these isolates to identify what type of antimicrobial resistance or virulence genes uh, there in the these different Listeria types. So we used ResFinder to do that analysis, and there we identify 5 different antimicrobial resistant genes across all samples, except one sample. The fosfomycin was identified in all the isolates. So other than fosfomycin, there are 4 other antimicrobial genes been identified, and all these antimicrobial genes were reported in lineage 2 isolates. So when you look at the virulence genes, listericin gene was identified in lineage 1 isolates, and which indicate the presence of different pathogenic islands. And same goes with VIP gene in lineage 2, which indicate the presence of pathogenic island 1 to 4. So the take-home message is like we try to illustrate, like using different tools, we try to analyze the prevalence of various serovars and sequence type and microbial resistance in Listeria species in food and food processing units in Northern Ireland, and England. And our study findings actually establish a kind of a historical record for Listeria species, especially in Northern Ireland, but it opened up further investigations uh, towards epidemiology of Listeria in the country. And also this study demonstrates how valuable is data sharing is, because all the isolates came from England and Ireland. We actually gathered from public domain. So this actually shows the power of data sharing, how it can use in collaborative analysis to analyze and characterize these pathogens. And taking everything together, actually, this actually can be fed into national surveillance data repositories across UK and European Union. And this information can be used in detecting foodborne pathogens rapidly and their antimicrobial resistance and also their transmission pathways. So with that, I would like to acknowledge our team in AFBI, in TB Molecular and Genomics Unit, and our Pathogen Genomics Unit for their guidance and sequencing and completing all the analysis. And also Food Micro Unit for collaborating with us for providing their previous data set, and we are actually working on a manuscript to publish the results. And also we would like to acknowledge the PathSafe project, which is funded by Food Standard Agency as HM Treasury in England. And we are collaborating with Dr. Ozan from LHS, London School of Hygiene Tropical Medicine, especially for the second part of the study. And I would like to thank the data collaborators, contributors in NCBI EMBL for sharing data this kind of research purposes. And finally, I would like to thank Safe Food Kit for giving us the opportunity to present our study findings. So if you have any questions, I would happy to answer during the Q&A session. And with that, I would like to hand it over to Nick. Thank you very much.
Okay, thank you, Purnika. Hopefully everyone can hear me and can see my screen.
All good.
What I'd like to do is, I suppose, bring that into the context of a food business and how whole genome sequencing can help a food business. So what I'm going to go through, maybe building on what Purnika has said, what sequencing is and why it's important, then how it can be used by food businesses and give some case examples, the practical application, and then talk a little about some of the realities of its implementation and some of the challenges and opportunities. So thanks to Linda for introducing me earlier. I'm head of food safety and quality with Dawn Farm Foods. We produce cooked and fermented meat and plant-based ingredients, but our ingredients are going into somebody else's products, whether that be a sandwich, a pizza, ready meal, or a food to go within food service or food manufacturing. And we see our job as protecting people's brands. Everything we make is ready to eat, and when you multiply up the number of portions we make, we're making many, many millions of portions of ready-to-eat food a week. So that's what we need to protect through our food safety management system. And I suppose just to put it into context, there are many things which will influence the safety and authenticity of products, whether it be the cost of living or weather patterns. And I suppose very, very relevant to this technology are foodborne outbreaks and animal diseases. So anything that we can use that helps to ensure the safety of our products, then we will employ that within our food safety management system. So why have we all heard of whole genome sequencing? Because it can so quickly and precisely fingerprint foodborne pathogens, it has been a transformative technology within public health. And that's a statistic at the top from the Food and Drug Administration in the US, that in the sort of 18 or so years of between the end of the 90s and the mid 2000s in the United States, subtyping helped to detect 25 times as many outbreaks. But importantly, the average patient case count fell from 69 to 4. So many, many more outbreaks detected, but each of them smaller because they're being detected more quickly. This technology, much like the space race in the 1960s, drove technology development. So the race to sequence the human genome developed many of the technologies that are now regularly employed in laboratories around the world for medical and foodborne pathogen surveillance. And at that time, what cost $3 billion, it can now be done for $100 or a couple of $100. Most public health authorities around the world have transitioned to whole genome sequencing for pathogen tracking. So in the USA, it was around 2013. In Europe, more kind of between 2015 and 2020. And another good example of how it's been used was during the COVID-19 pandemic. So the COVID-19 sequence was actually published within days publicly online, and then online open databases were used to track its behavior and its spread around the world. So it was very much a pandemic that utilized the benefit of whole genome sequencing, both for tracking it around the world and for the development of vaccines and medications. It's worth saying as well that in the United States, the Food and Drug Administration have done a cost-benefit analysis of the implementation of whole genome sequencing. And they judged that between 2013 and 2019, for a cost to public health agency of around $22 million, it has actually saved $500 million in terms of public health. So through less mortality and morbidity. So they're very much demonstrating a cost benefit of implementing the technology. And this is a quote from Frank Yanis, who was in the Food and Drug Administration himself, ex-Walmart, a well-seasoned veteran within industry and within the regulatory authorities. He said, what's the number one thing that's advanced food safety in the 21st century? And his favorite answer was these three short letters, WGS. But that's very much looking at it from a public health perspective. And there's plenty of examples of how whole genome sequencing has helped to track and trace foodborne outbreaks. So just to give some examples, I mean, at the moment, we have a prepared meals outbreak going on in the United States. So that was detected through whole genome sequencing, matching it to prepared pasta meals and actually linking it back then to the prepared pasta from a supplier and tracking illnesses all the way back to the start of 2024 through the summer of 2025. There's quite an interesting one, how an unusual serotype of Salmonella that's caused something like 600 illnesses since 2011. And researchers have looked back through the data to see that it's a very tight cluster of strains that have been making people sick across Europe seasonally, associated with tomatoes from a particular growing region in the south of Europe. So these are two examples really of where sequencing is connecting together illnesses that maybe have happened over a long period of time that previously would have previously gone unnoticed. And the last example I'd just like to give was a big recall in the United States 3 years ago, where Abbott recalled infant formula due to an association with Chronobacter cases. But what's kind of interesting from that one is actually that in retrospect, WGS actually separated the samples from the sick infants from Chronobacter that was found in the environment. And it's interesting to see the Abbott website and how they how they feel that the recall was unjustified when you actually look at the sequencing data. But despite the use of the technology, some illnesses are still increasing. So Purnika spoke about listeria there. So the trend in listeriosis in Europe and America and other developed countries has actually been increasing since the 2000s, and the same with STEC. So there is opportunity yet for these technologies to actually start making an impact. And we'll just pause for a moment on what the technology is. Purnika went through this, but kind of from a layman's point of view, you start with an isolate. So whether that's Salmonella or Listeria or Chronobacter, the DNA is then extracted from the cells and then goes into a sequencing instrument. And this is where the really clever chemistry goes on to replicate the DNA sequence and multiply it and actually turn it then into data. And that's where then the bioinformaticians come in, because what comes out of the sequencing instrument is ordered but very much unintelligible data. So bioinformaticians, sort of cross between microbiologists and computer scientists, they use software tools then to turn that data into information. And that can give you, I suppose, a set of different pieces of information about different isolates. So whether they are related, and what you see there is a sort of general phylogeny of salmonellas from surveillance of meat supply, showing the sort of routine serotypes that you might find in meat supply. But it can also deeply characterize the isolate, give you the serotype, the sequence type, the core genome type. So really drilling down into the taxonomy of the organization. But it can also give you genetic features that maybe are associated with some particular behavior. So whether or not the organism has genes that mark particular virulence or sickness causing capability, or whether it's multi-drug resistant, or maybe whether it's got determinants that make it more tolerant to food processing environments and to disinfectants and things like heavy metals. So there's a whole suite of pieces of information that you will get from the sequence which fall out of that bioinformatics, where previously they would have had to go through separate sort of laborious methods within a laboratory to find out those pieces of information. So for the food business, how can you use it? Well, you can use it for track and tracing contaminants, whether that's tracking salmonella within a meat supply ingredient chain, or whether it's listeria in a prepared ingredients chain. It can enhance the environmental monitoring by giving you information about each isolate that may be found in the environment and its connectedness with other isolates or made with the ingredient supply chain. It can be used for risk assessment, which I'll touch on shortly. Not to forget that it has a role in authenticity as well. I mean, DNA species surveillance is, I suppose, another branch of genetic subtyping, seeing what species are present in a particular food sample. And what is developing is the concept of precision food safety. So in parallel with precision medicine, the concept that you can actually tailor therapies to the particular individual. So the promise is that with sequencing, there's this opportunity to precisely tailor the intervention to the particular characteristics of the bug that you might find within the environment. And finally, again, same technology, but used slightly differently, the microbiome. So rather than starting with an isolate, actually looking at all the different organisms that might be within a particular sample, and then mapping those as you go through the shelf life of a product, and being able to build predictive tools using artificial intelligence so that you can predict the safety of recipes. So to give a couple of examples quickly, this is a real life example from some years ago of using subtyping where a ready-to-eat ingredient was being purchased with certs of analysis and positive release into the plant. And a new delivery of the ingredient tested positive for salmonella, so was rejected. But very shortly afterwards, a product that was being processed tested positive for salmonella. But what subtyping allowed, along with a rapid traceback of the components through serotyping by the Central Veterinary Research Laboratory in Back Western and fingerprinting by our collaborators in UCD, comparing that then to results from the seasoning components. You can see there at the bottom the patterns that come out of an older technology called PFGE, which showed a precise match between salmonella in the seasoning and salmonella within the work in progress, the product. So it absolutely wrapped up within a week or two the precise cause of the contamination within the factory. And also because that ingredient was used in other products, in other parts of the food sector, it actually helped to prevent an outbreak. So very much an example of subtyping in action. The other example I just wanted to give was of risk assessment. So this is actually a graphic from a fantastic piece of work by French researchers in about 10 years ago. So up the left-hand axis is the frequency of different listeria clones within clinical setting, within illness. And along the bottom axis is the frequency of listeria clones within food isolates. And what you see is that there are clones with boxes in red, which are disproportionately associated with food illness rather than with foods. And there are clones that are disproportionately associated with foods rather than with illness. And the researchers came up with these concepts of hypervirulent clones, those that are very, very capable of causing disease and more often seen in clinical cases, and hypovirulent, so relatively benign listerias that are more adapted to the food environment and are more commonly seen in foods, but don't typically cause illness. And so it's sort of an example of where, drilling deeper into the characterization of the organisms gives you some indication of its potential risk from a public health point of view. Not taking away, of course, that all Listeria monocytogenes are potentially virulent. But this concept has been picked up now by the FAO and the WHO, particularly when in emerging economies where it may not be appropriate to withdraw a food if it has a very low level of contamination by one of these food-associated clones. So, you know, this concept of knowing the enemy and using that for risk assessment. Some of the realities of this, you know, there is a cost to it. It's you know, it's of the order of hundreds of euros to get something sequenced, obviously on top of it having already been isolated by a laboratory. But that insight has to be balanced against, or that cost has to be balanced against the potential benefits when you've got product batches that may have values of tens or even hundreds of thousands of euros. So if it's put into the context of an overall lower reject rate, then the benefit is there. But it does take a little time. So it doesn't necessarily change the heat of an investigation if you find a pathogen in the environment and need to make corrective actions. But over time, it gives you insight into where it may have come from. So whether it's a different subtype that keeps coming in, which suggests that your hygiene barriers aren't working correctly, or whether it's a common persistent strain, which suggests that your equipment or your fabric is harboring a niche. So that has insight over time into the way you construct your factory, the way you construct your utilities, the way you do the hygienic design of your equipment. So over time, it can give benefits as well as that relatedness in the short term. Sensitive to precision, it is an extremely precise technology where essentially it gives you a forensic match to any other isolate that has the same fingerprint. And you do need to be aware of I suppose, the precision of that data that you're generating in-house if you start using this technology. So from that perspective, the security of the data is absolutely critical between you and the laboratory who is developing that data, because in the wrong hands, that can be you know, it can be misused because of its precision. Purnika actually commented on legislation and this new EU regulation that will come into force next year, which is primarily aimed at regulatory authorities having to use whole genome sequencing throughout Europe for outbreak investigations and to share the data between them. But it also has a clause which is relevant to industry, because it requires that food businesses will submit sequence data to the competent authority if there is a suspicion of their being involved in an outbreak. So look, it's something to be aware of. It may make certain businesses think twice about implementing the technology, but it also has the opportunity to actually separate you from an incident if you have your own database which demonstrates that there's no connection. And finally, you know, there are alternatives. I mean, we do talk about whole genome sequencing, but one of the examples I gave there was with a previous generation subtyping technique. There are other techniques available like ribotyping or sequence typing, or certain technology providers have platforms that will very rapidly characterize an organism, not all the way down to the sequence level, but to slightly higher level. You know, and they can be quicker and less costly and still give huge value in terms of understanding the connectedness between different things that you might find within your environment. So to summarize, we see that whole genome sequencing is a technology that is really helpful in terms of keeping people safe, by keeping the products safe, by keeping reputations intact, by ensuring the integrity and authenticity of the food products that you're providing, building trust. And what we have always found is that deeper knowledge about any particular detection gives security to the situation, because you know more about it. You've got more risk assessment opportunity. You have a database of information to look back through to understand whether something is connected or not. So thank you for listening. And you know, if there are some questions, happy to join the chat at the end. And I'll now hand over to John.
Thank you, Nick. Hopefully people can see my screen.
Thanks,
Linda,
John.
maybe you can.
All good, thank you.
Okay, all good. So, okay. Thank you very much, Nick and Purnika before me. The difficult is always when you're the third speaker, there might be a risk of some overlaps, but I think that will do no harm. There'll be a little bit of overlap. I'm John Donaghy. As Linda had mentioned at the beginning, I'm the global head of food safety for Nestle, and I always put, when I go to these sort of presentations, I put a caveat that to put it in perspective, Nestle is a large multinational, uh, you know, um, organization, let's say, and I manage 350 plants across 80 countries. So sometimes when people ask me, why do you implement whole genome sequencing and why do you do it in-house? I always say we can have the potential ammunition to work with across that number of factories, operating from infant formula through to pet food and every type of food business you can think of in between. So there is always let's say, some issue where you want to investigate from a microbial safety perspective. And I think whole genome sequencing has become one of our go-to tools, to investigate issues. So today, what I want to take you through is a little bit to the public health perspective at the beginning. Then I will take you through why and how we do it in-house. I will give you some concrete examples of where we have actually applied it. And hopefully I'll demonstrate some of the economic benefits from a food industry perspective. Nick talked about the benefits from a public health perspective that FDA have done these calculations. I can give you many examples of where we have shown the financial and economic benefit by actually doing this in-house and where it has brought benefits for us. So to begin with, as has been mentioned previously, we have a new set of tools now, the food safety microbiologists. Whole genome sequencing is the one we will talk about today. There also is metagenomics, and sometimes there's a little bit of confusion between microbiomic studies and metagenomic studies and whole genome sequencing. And in essence, you take a sample. On the left-hand side of this screen, you can see it could be a raw material, it could be a factory environmental sample, it could be a finished product sample, or it could be a clinical sample, and you get an isolate, so an isolate of a pathogen from that, and you sequence it. On the other hand, metagenomics, which is less mature, I would say, and less used in the food sector, involves just extracting the whole DNA out of any of those samples, and then doing a profile, like in a microbial ecology, of what the organisms may actually be in that particular sample. But today I'm going to stick with a whole genome sequencing, and talk about where we can actually apply that. As I said, has become the go-to tool within the public health sector, and I think I could hardly name a foodborne outbreak that has not been investigated using whole genome sequencing in the last probably 10 to 12 years, starting from about 2010-12 onwards. Basically, most foodborne outbreaks are investigated by whole genome sequencing, and either it's in developing countries, which can do it themselves, or if it's a developing country, someone else will do it for them. Nick just talked about some of the recent ones, and I reiterate that one as well. This is Salmonella strascona, which is a multi-country, multi-year outbreak, which through the use of whole genome sequencing has now been traced back to tomatoes from Sicily in Italy. Anyone who watches the international food safety news, you will see that there's been multiple recalls relating to salmonella in pistachios since about August onwards, and again the salmonellas that have been isolated in the pistachio nuts and products have been traced back to nuts or pistachios emanating from Iran. So again, this is through border checks, controls into the US or into Canada, and what the people have been getting sick with. So again, this demonstrates the precision and the trace back or source attribution that it can give you the benefit of. And then there's a few other historical cases I just thought maybe closer to home. One was in the UK hospitals a number of years ago, where patients in a hospital became sick with listeriosis, and they traced this back to a sandwich maker who was providing sandwiches to the hospital, and they were able to detect the same Listeria monocytogenes was whole genome sequence type in the sandwiches that the people were actually getting sick with. And they even went further than that, and they analysed the components of the sandwiches, and they found that the same strain was present in the meat manufacturer who was providing the meat for the sandwiches. So again, that demonstrates how whole genome sequencing can be used for this traceback or source attribution scenario. And the same happened for Greenyard. This was a large case, and it's actually created a lot of tension in the EU, and this was related to frozen corn. This took a long time to solve across multiple countries in Europe between 2015 and 2018, and there was, as you see, 9 deaths and 47 cases associated with this. But it transpired that this actually the same strain of Listeria monocytogenes that was present in the environment of the factory producing the corn was the same as the strains that the people were getting sick with throughout the different countries throughout Europe. So clearly, as a public health source attribution tool, whole genome sequencing has proven to be invaluable. And generally, when these outbreaks happen, and this is maybe this demonstrated in your talk in September, generally when this happens, there's 3 bits of the jigsaw that needs. There is the illness, so you have patients who are sick. They are probably shedding the pathogen in their stool samples, etc., or perhaps even in blood samples, and then those are sequenced. Then generally the public health professionals will ask them, "What have you ate in the last 2 to 3 weeks prior to you getting sick?" Or they might ask, if they have a suspect food, they may ask, "Have you eaten this particular product in the recent 1 to 2 weeks before you've got ill?" And then they will do, if they're able to find this material, the foodstuffs, that if it's not perishable, they're able to recover it. For example, if it's a frozen product, then they will try to isolate the organism from that foodstuff, and then they will whole genome sequence it. And if it matches exactly the strain that was present in the patients, then that, coupled with the epidemiology, more or less makes it a closed case. So you need the epidemiology, but you've got the whole genome sequencing as the forensic tool. So that's really what happens in a public health let's say scenario with regards illness and foodborne illness source tracking or source attribution. But I am a strong believer that, if this tool is strongly used from a source attribution for these outbreak scenarios, then surely the food industry can also benefit from using this. And I say this is to get ahead of the tool. You imagine you're able to apply this tool to solve some of the problems that you may face in your manufacturing plants. And typically the questions that you will have from a food manufacturing perspective, and these are the questions I deal with every day across our plants, is the strain that we're seeing that we may find in an environment or in a product contact surface, or maybe even in a finished product or a raw material, is it a tourist or is it a resident? Is it something that comes in occasionally, and then we do our cleaning, sanitation, it disappears, you might get the same material comes in at a later stage, another delivery, it comes in with a delivery. So in other words, it's a tourist, but you're able to manage it through your cleaning, sanitation process. The bigger challenge is if it is a resident, if you realize this same strain is pretty widely spread in various parts of my manufacturing facility. It could be in drains, it could be in hidden dead ends, it could be hard to reach places. That is a bigger problem to deal with. The other thing is you might want to understand, do I find the same strain? I might find Listeria in one part of the factory, Listeria monocytogenes, and find it in another part of the factory. But is it the same? By traditional serotyping, this does not tell me a great deal, but the precision that whole genome sequencing offers tells me exactly is the same one moving from one area to another area, and so helps with my corrective actions. The other big question, which we get quite often, is when you send your sample from a factory and you send it to a laboratory to test, you will and they find a positive finding, whatever pathogen it may be. Generally, the food manufacturer, the first question they will ask is, "Are you sure?" Is it a false positive? And this can be an apprehensive situation even for the laboratories to make sure, are we sure? So, and this is where I will show some examples of where laboratory cross-contamination, can be easily solved by WGS as well. And then the other part is, as most food manufacturers, you are buying lots of different raw materials, and sometimes the pathogen, even though you might have a certificate of analysis, you might do hold and release of your raw material coming in, still you might have a pathogen could come in with some raw material and contaminate your environment. So it's important to understand, am I getting it from my supplier as well? So these are all typical questions which we pose to ourselves within Nestle. And remember, within Nestle, we do all of our 95% of our testing all in our own laboratories. So if we find a lab cross-contamination, it's our own kitchen. And we do about 4 million microbiological analysis per year, just to put it in perspective. So these are what I call some of the benefits that the whole genome sequencing can actually bring to a food manufacturing business. Nick, I think, mentioned, and we've all heard the examples of, it gives us a better insight because of the precision that it gives it. It gives us a better insight for root cause analysis. And as a result of that, it actually means that our corrections and our corrective actions are actually more sustainable. Because quite often, you can find a pathogen in an environment of your factory. You make a change. You think you know what the root cause is from a microbiological perspective, but 3 weeks later, you have the same situation. So clearly, you did not get to the exact root cause and you did not do the correct correction or corrective actions. So with whole genome sequencing, with the precision that it gives you, you can really focus in on where the problem is within the processing plant. And the one on the bottom right hand side of this screen is contributing to the robust verification analytical process. And this is what I will give an example of, a laboratory cross-contamination situation where whole genome sequencing can also help and has helped us in a large number of cases. And by doing this better root cause analysis using WGS and better corrective actions, you end up reducing waste as well, and you reduce analytical costs. Because typically in our factories, we will have a regime to escalate when we have a finding. And if we have a finding, and you imagine you had a finding and there was a laboratory false positive, and you end up disposing of a lot of product. You end up doing a lot of escalated testing, a lot of investigative testing, and it was a laboratory cross-contamination. That was a lot of waste you have generated. So with whole genome sequencing, you can really determine if it is a laboratory cross-contamination or not. And then it also helps build the trust in your supplier management program, because quite often we work quite closely with our suppliers, and if we have a finding in raw material coming into our factory of a pathogen, we can work with our supplier, ask them to swab and test more in their factory, and then we eliminate their problem by us testing at our side. And the other big important point is environmental monitoring improvement. All manufacturing plants will have an environmental monitoring program where they do lots of swabbing in different parts of the factory, product contact surfaces, a little bit further from product contact surface, and various areas within the processing plant. If you know exactly what organism you have, and if you know if it is similar to one that's found close by, then you can redirect your environmental monitoring program. There could be areas you have to increase the number of routine testings in, or increase the frequency because of the information WGS can actually give you. So now I'd like to take you through some examples, really concrete examples, or where we have actually applied it in our plants. Or maybe just before that, I'll just to highlight what Nick had said, and Purnika had said as well. This is typically our process. You have a food factory. You take an isolate, so we send it to our laboratory. Our laboratory, if they find a salmonella or or a Listeria monocytogenes, we will do what we call the wet laboratory side, the sequencing, and then we'll do the bioinformatics. Now, in Nestle, we do this ourselves. So different industries approach this in different ways. First, they determine, are we going to do whole genome sequencing? If they decide that they will do it, do you build the infrastructure yourself? Now, there's a few large manufacturers like ourselves who actually have built this capability in-house to do WGS. Others use commercial providers, and there's enough commercial providers out there now across many of the diagnostic type companies, and some of them use academic partners to do their sequencing. The important thing as well is, do you want to build a database? So if you sequence your isolates of pathogens, you can actually build a database, and this can actually be very beneficial. If you have a case, in 2 years' time, you can look back and say, did I find this same strain previously in the same plant? Because then you have this database of sequenced pathogens. But this is a little bit sensitive around the building of these databases and the sharing of information from these databases. In Nestle, we do build our database, based on the sequencing that we've been doing over the last 10 years. So as I say, we do it in-house, and we've seen the application and the uptake of it internally across our plants globally has been increasing over the last 10 years. We started doing whole genome sequencing in about 2014. Doing it for real after we established all our pipelines, and our pipelines are very much aligned with the FDA type pipelines. So now it's nearly a situation where I'm trying to stop factories doing too much, because they see the real benefits of it, either for the root cause investigations within the factory or within the laboratories. And the typical pathogens that we will look for and that we will sequence will be Salmonella, there'll be Listeria monocytogenes, and Chronobacter, because we're, I think, one of the largest infant formula manufacturers in the world. So Chronobacter is a pathogen of concern for us as well. But we have done ad hoc whole genome sequencing on other organisms like Bacillus cereus, Staphylococcus aureus, and even Clostridium botulinum, or Clostridia species rather, to determine if it was botulinum or not. So that's a little bit our internal kitchen, as it were. So now, as I mentioned, I will go through a couple of examples of where we've actually applied. This was an example of a food factory, dry culinary factory. We had collected a number of Salmonella mandaka strains, and then we had a suspicion, where could this be coming from? When we do it by serology, so typical serology that doesn't have the precision of whole genome sequencing, and we found that they all looked the same. So all the isolates that we got, they all showed up as Salmonella mandaka when we did just normal serotype. When we did this by whole genome sequencing, and this is the precision that Nick talked about, we're able to demonstrate that they were very, very distinct clusters of Salmonella mandaka. And this has allowed us then to go to individual suppliers and say, look, we've got a unique Salmonella mandaka. We're wondering if it maybe came from you. We went to some of those plants. We surveyed our suppliers' plants, and we were able to find that the milk-based powder was manufacturer was the person who was selling us our most prominent Salmonella mandaka that we were finding in our environment. Prior to that, using the serology, we were guessing. Most people are thinking it's probably the garlic powder, for example, because it could be prone to Salmonella, a dry, agricultural-based powder. But it turned out, by using the precision of whole genome sequencing, it allowed us to trace this back to a particular supplier of milk powder. Another example related to a chilled and frozen culinary factory. Again, lots of raw materials. You imagine a chilled and frozen factory, you get a lot of agricultural raw material. So we had a collection of Listeria monocytogenes, and the question was, where are they coming from? Are they the tourist or are they the resident? So we did a lot of sequencing of all the strains we had in that particular factory, and it very clearly showed that a large percentage of those isolates were coming from one piece of equipment that we had in that particular plant. And so that really narrowed down our root cause analysis. And we tried many actions, I must say, in that particular plant to try to clean and sanitize that piece of equipment. and we were defeated, and we end up having to remove the equipment completely and install a brand new, better hygienically designed piece of equipment, because Listeria monocytogenes is notoriously difficult to eliminate from chilled and frozen factories. And but this whole genome sequencing allowed us to say our biggest problem is the residents that are in this machine, and they all seem to be the same. Prior to that, we had no idea. Was it just continual intake of Listeria monocytogenes from different raw materials? So again, this allowed us to make a proper corrective action, i.e. replace the piece of equipment. Another way we've deployed whole genome sequencing has been doing, you may have heard of, swabathons. This is very popular with the FDA. They come into your factory, they spend 3 weeks in your factory, and they just take swabs from everywhere. Roofs, ceilings, floors, contact surfaces, drains, etc. And when they find, then they sequence. We decide to do this ourselves in some plants. And this example on the right-hand side is an example of, again, of a chilled frozen factory where we do Listeria swabathon. So we go in and we take hundreds of samples from all areas, not your routine sample areas, but even other abnormal areas, like say deep in drains, etc. And the red ones indicates the Listeria monocytogenes that we found in that particular plant as a result of the swabathon. Not rarely, or it was rarely detected as part of our routine environmental monitoring, but with the swabathon we identified where we're having these different Listeria monocytogenes. The question then was, are they the same or are they different? So we had about 100 isolates and we said, are they the same or are they different? 90% of those isolates were all exactly the same by whole genome sequencing. And they were all, when we did the investigation, we were able to trace these trolleys, which were moving from one part of the factory to another. So as people moved the trolleys around from one part of the factory to the other, it was clear they were moving the Listeria, this unique Listeria monocytogenes, with the trolleys. So the simple corrective action was, we have two separate sets of trolleys that never move from one area, a particular zone, to another zone. An example of whereby this swabathon allowed us to get to the root cause, and it allowed us to implement corrective actions through the changing out the trolleys. So moving away a little bit from inside the factory environment and where whole genome sequencing has offered us benefits, and it's well known that you can have laboratory false positives or cross-contamination in laboratory. This is not a phenomenon that's new, but what is new is with whole genome sequencing, you actually can prove that there is cross-contamination in a laboratory, and where those cross-contamination can come from could be your positive control that you use in your particular assay. It could be the proficiency test sample that you've actually been using, and somehow it contaminates the sample or cross-contaminates the sample you're testing. Or it could be a previous test sample that you used for another client that cross-contaminated your sample. And we have discovered all three of those or sometimes in our own laboratories and sometimes in third-party laboratories. And I'm going to give you one example to illustrate this, and it illustrates also the economic benefits of doing WGS for us. We had a factory in a country, I will not name the country, and for this particular country we produce a certain product. This product has to be tested by the regulatory authority in order to be exported to other countries as part of intermarket supply. So it goes to the government laboratory. The government laboratory tests our sample and they say, we have found positive Salmonella typhimurium. So that would normally mean we should dispose of the product. We cannot export the product. But they themselves said, look, this is unusual. We have never had Salmonella from your plant before. Maybe you want to do whole genome sequencing. And so we agreed with the laboratory to do whole genome sequencing. So we said, what's your positive control that you use? And they said, our positive control is Salmonella tranorea, which is a rare Salmonella type, and that was okay. So we sequenced the Salmonella typhimurium that they purportedly found in our product, and we sequenced tranora. They were, as they were they're supposed to be. The Salmonella typhimurium was Salmonella typhimurium. The Salmonella tranorea was Salmonella tranorea. However, the unusual thing about this situation is we had our own laboratory in the same country, and the Salmonella typhimurium that they found in our product was the exact same as, because we have a database of the positive controls we use in our laboratories, it was exact same as a positive control in our own laboratory. So this is where then you start to put on your Sherlock Holmes hat and say, how could this possibly happen? And we had many hypotheses as to what could have happened, including could our laboratory send the containers for sampling from their laboratory to the factory filled finished product into those, and the containers were contaminated. So we had all sorts of theories. Then luckily we had someone who was very vigilant and went through the COAs from the government laboratory over the last 6 months, and they discovered 3 months earlier that they actually used typhimurium as a positive control, not Trinoria on that particular occasion. So we asked them this situation, and they said yes. In that case, we had no Salmonella Trinoria growing, so we used typhimurium as a positive control. So we said, can we sequence that? We sequenced that one, and it was the exact same as the Salmonella typhimurium that they had got in our product, and the exact same as the one we'd found in that we had used as a positive control in our laboratory. So clearly, positive control, cross-contamination. This particular event saved us 3 million of the currency of the country we were in. So when we talk about implementing, when I have to justify return on investment of implementing whole genome sequencing in Nestle, I can just quote this one example is sufficient to pay for the whole implementation of the program and the program we have running. So this, and this is not an isolated case. We have had situations like this before. So clearly, I've tried to give you some, let's say, real life industrial examples, but in the bigger scheme of things, I mentioned earlier, databases are important. And one of the largest databases that is available is called Genome Tracker. And this is the database of pathogens that is established and maintained by the FDA. And many laboratories around the world, like Public Health England, submit samples to this. Many government laboratories submit samples to this. When the FDA inspectors go to their visits to their plants, they will take swabs. If they find pathogens, they will sequence them and put them into Genome Tracker. So what you see in this middle graph is actually almost, to date, there's almost 2 million sequences of different pathogens, Salmonella, E. coli, even if Shigella in there, Listeria, some Chronobacter in that. And it's also populated by isolates from the CDC, so clinical isolates. So if something someone gets sick, they have the isolate, the first thing that is done is they will go and access that database and see, have we seen this particular whole genome sequence type before? And this adds a little bit of a worry to factories in the US in particular, because if they come in your factory, the FDA, they swab your factory and they find a pathogen, then they go back to the database and they say, okay, we have found it, and this came from say it matches a clinical sample from 3 years ago in a certain state in the US. This is where industry gets a little bit anxious, and certainly the legal team gets a little bit anxious in these scenarios. But you can see the benefits from a sort of a global perspective and public health perspective in terms of tracing outbreaks across the world, not only within a blocked nation, but also across the world. And Europe is going the same direction. I think, as Nick has mentioned, that there's the new legislation coming. But prior to the new legislation coming, Europe has been building what we call this EFSA One Health WGS system portal. So they are building a database. So any strains that are shared across member states will be populated into this database. So it's more or less a replica of what the Genome Tracker is doing in the US. So you'll have a large database of all sequences of pathogens available. And often one of the arguments that I hear about WGS, so they say, oh, the technology is not that mature, the pipelines are not that mature, there can be variability, etc. Do I believe the technology? That argument has now gone, I'm afraid, because there's an ISO standard on how to do WGS for the wet chemistry part and for the dry lab, the wet lab part and the dry bioinformatics. So there is an ISO standard which majority of people should and and should be following. So the technical aspect is really gone in terms of an argument for us way not to use it. But of course, now you have this legislation coming in Europe, and the legislation by its intention was that so that member states would share isolates. So you imagine you have an outbreak in country X and you suspect it was from a product in country Y, but country Y does not want to share their isolates. Now, as part of the new legislation, countries will be obliged to or mandated to share isolates across member states. But as Nick has said, the piece that makes industry slightly concerned in the European legislation is this bit where it says where available. So if you are one of the companies doing regularly whole genome sequencing, then you have the material available. And if there's a suspected outbreak and it's one of your products, then you will be obliged to share those sequences with through your competent authority. So sometimes when I present whole genome sequencing, I have a title called Applications and Apprehensions. So you can see there is a little bit of apprehension about using it still within the industry. Laboratories in particular are certainly know the power of whole genome sequencing now for proving lab cross-contamination, and we know a lot of commercial laboratories now have changed the ways of working in light of the fact that WGS is available, and you could be subject to a claim by a manufacturer if you have had a false positive, for example. So there is still some apprehension around the use of WGS. So just to conclude, I say it is a mature technology. If I wrap around to my very first slide, metagenomics is a less mature technology. Whole genome sequencing is now a very mature technology, well-established pipelines for all pathogens, Chronobacter, Listeria, S-Tech, etc. And it is a go-to tool. Any foodborne illness will now be investigated by whole genome sequencing. Many of you may have been watching the news recently, and there's the large infant botulinum outbreak in the US. And I can tell you those strains have been sequenced already that they've found in unopened cans. That's how fast it can be done. And they are the same strains that were found in patients. So this is where the links can be made now very fast. So it is a go-to tool. But I still maintain that industry can benefit greatly. Even you saw some of the examples I've given from the use of the tool itself and get ahead of the curve. If we can use WGS so that you're not being asked by an authority to coming with a whole genome sequencing, you can actually say to yourself, look, I'm using the best available tools to try to solve my problems that I may have in my manufacturing facilities and get to a good corrective actions. So, and I think I've also demonstrated the economic benefits of using it as well with that one particular example. But I have many examples like that where we've had definitely beneficial economics or outcomes from using WGS. So with that, I will wrap up, and I think we will probably then all come back together for Q&A.
That's great. Thank you, everybody. Thank you to the three speakers for their excellent presentations. And we have a number of questions in the Q&A box. So I'm just going to go through those. I don't think people have indicated, who they want to direct it to. So I will sort of leave it open maybe to the presenters to see whoever is comfortable taking the various questions. And the first question there is, can the technology be used to fingerprint any organism?
I can jump in and take that one. Sorry,
yeah,
jumping ahead
perfect.
of you, John. I think as John has explained, yes, it can. Any organism that's been isolated can be sequenced. So, we've talked about Listeria, Salmonella, Chronobacter, but it's equally applicable to S-Tech, Campylobacter, Clostridia, and also to other organisms like viruses and parasites. So, it's really if there's an isolate of an organism, then sequencing can be used to identify it and to find relatedness.
Yeah, totally agree,
Great,
Nick.
thank you,
As I say, we have used it on, outside of the normal pathogens, we have used it on a number of other bacteria, just even to demonstrate that they didn't have certain virulence factors. We have sequenced Clostridia strains, just to demonstrate they didn't have toxin-producing genes. which sometimes because it's very difficult, let's say, to isolate Clostridium botulinum, because it's a bioterrorist type agent. So, but you can sequence, if you've got a Clostridia, you can sequence it, and then you show that it doesn't have certain genes by whole genome sequencing instead of PCR.
I can add a little bit more into the conversation, especially when it's come to virus, like bacterial phages and fungal infections. So it will be quite difficult in that area because the databases for identification of different organisms can be a little bit tricky because the databases are not that comprehensive at the minute. So, but it is good to carry out whole genome sequencing in that area in order to support fungal and viral characterizations.
Great. Thank you, Purnika. We'll move on to the next question, which is around the current EU legislation that's been referred to in relation to listeria testing and the use of genomic analysis. So does anyone want to take that, the changes that are coming in to the listeria legislation?
I think the previous
just maybe brief.
legislation.
Yeah. No, go ahead, Purnika.
Yeah, I think the previous, sorry about that. Previous legislation is mainly around like microbial confirmation using different other techniques like MLST or serotyping. So whole genome sequencing is introduced as a kind of a requirement now, just to make sure like all the ready to eat meat products or like food products are free of different pathogens. So that was more like the addition into it, I think. So this will be come into effect in next year, July, as far as I'm aware.
There's probably two elements to it. One is the new listeria legislation, and then there's the whole genome sequencing legislation. So not to maybe you confuse the both. The new listeria legislation is really about now, because in the past you used to have no growth foods or growth foods, and depending on whether it's a growth food or a no growth food, your microbiological criteria was different. So it could be less than 100 CFU per gram, or it could be absence in 25 grams. But there was a court case which then found a loophole in the previous European legislation, where someone released a product as absence in 25 grams, but then the product was a growth product and it grew to levels that caused illnesses. So now the new legislation is really all about showing through challenge tests that you maintain the same levels if it's absence 25 grams or less than 100 to the end of shelf life. So that's a very much a listeria driven legislation. The whole genome sequencing legislation is just about outbreak investigations and mandating member states to share isolates, and the clause that Nick and I mentioned about where applicable industry must share as well. So both are they're slightly different legislation. We could have a whole webinar about the listeria legislation. I'm sure there's been many done before, but that's a slightly different one.
Great, thanks for that, John. And we are actually planning to have a webinar next year on the listeria legislation. So I think there will be a lot of interest in that as well, but obviously separate to the whole genome sequencing, as you said. And now there's another question about how should a food business determine when to apply whole genome sequencing? Is it routine monitoring, outbreak investigation? Like, where's the best kind of use of the technology? I think is what's being asked here.
I'll jump in to suppose give a comment on that. There's no particular place to start, but I suppose probably most businesses who use it will start during an investigation, because that's where you'll demonstrate the real power of it. So, but look, it'd be better to start in peacetime and use it during routine monitoring. Just sequence a selection of isolates over a period of time and see what you learn, so that when you get to an investigation, you're ready, prepared for the types of results that you will get.
Yeah, and that's a little bit the same from our side. You notice on my slide, I say we use it as a routine of investigation, not as routine. It's a routine of investigation. And of course, we will have our own criteria as to when we think serotyping is enough. If I think of salmonella. So if I say, look, serotyping will give you enough information, I don't advocate proceeding to WGS. And I will always ask our plants, what's your hypothesis? So if they say, look, I suspect it's lab cross-contamination, and there's a lot of product implicated, or maybe there's been a history before of laboratory poor GLP or whatever, then I will approve to do sequencing, and then I will ask to sequence the isolate that they supposedly have, the proficiency test isolate, the positive control they have in the factory, and any recent, if it's salmonella, that they've had in the factory. So those 4 isolates will be sent for sequencing to prove or disprove the hypothesis of laboratory contamination. But that's why every industry have to have some criteria, because, as I say, there's whereas when I began doing WGS in 2014, people, too expensive, I don't want to do it. Now it's a little bit the opposite. People they see the power of it, but now you have to tell them, look, it's expensive, yes, so don't be doing it on a routine basis. If there's something that it will prove and will allow you to go with corrective actions or help with a root cause, then proceed. But we are very selective as regards when we use it for an investigation.
Great. Perfect. Thank you, John and Nick, for answering that one. And the next one is, I suppose it's not too dissimilar, so how we can integrate WGS results into a food safety management system. So how would you use that, I suppose, to inform your HACCP or whatever system, your food safety management system you're following?
Well, Michael, maybe I have a jump at that. In any food safety management system, one of the key pillars is your analytical verification. That's a key pillar of any food safety management system. And you obviously have to have robust verification, as I said. So, then you decide what platforms you use for your normal testing. So this is let's say your analytical portfolio. So I test for salmonella, I test for listeria, whatever pathogen you test for, and whatever hygiene indicators you test for. Then the question is, do I supplement that with whole genome sequencing? And of course, then you can, and that's what we're talking about today. I would say we have WGS embedded in our analytical verification and diagnostic leg of food safety management system. That's how I would sort of sell it as part of that. And of course, the information that you glean from WGS can help inform, let's say, your hazard identification and hazard assessment part of a HACCP study. The front end of a HACCP study is hazard identification and hazard assessment. So you say, look, we regularly see these types of salmonella in this sort of raw material. And then that directs me a little bit how I try to manage that by getting a COA from my supplier, doing a lot more monitoring, etc. So yeah, it feeds a little bit back into the HACCP study, but it's more or less part of your suite of analytical verification tools.
Perfect.
Yeah, I'd add a little to that, as well as being embedded kind of in the investigative side. We would have it as part of our environmental and our ingredient monitoring programs, and really that's looking for connections rather than just it's there or it's not there. So building that database over time, as John said, so that you're getting further insight into programs that already exist. So sort of extending the technical content of those environmental and surveillance programs.
I think in general, it's built up a kind of a baseline. So what is in there in different food types and even in different regions. So that's actually fed into general surveillance programs.
Okay. Excellent. Thank you. And then there's a question about the in-house capacity that's needed, if you want to do whole genome sequencing in-house versus outsourcing it to specialized labs, and kind of what are the considerations there? Don't know who wants to take that.
I'll take it a little bit. As I said at the beginning, we're a little bit unique in that we're a giant. Nestle's a giant with so many plants, and you have so much ammunition for something like whole genome sequencing. If I was sitting and advising someone and they've got 10 plants, and the last time they saw salmonella was 10 years ago, I'm not going to build in-house infrastructure to do WGS. So I use a local partner, be it an academic or perhaps some of the commercial labs now that offer this service. And I think that's a little bit, you have to weigh up what are the benefits. One thing is, do I want to do it? Then if I want to do it, do I have enough ammunition for it? And then do I do it internally or do I do it with an external partner, like some of the commercial laboratories, or an academic partner? That's a little bit should be your sort of decision tree with regards, where and when you do it and who with. We can do it, and we have implemented it, and even we have extensive infrastructure of laboratories around the world, but we only do WGS in one centre location. So we have decided to do it, and we decided just to have it in one. It does present some logistical issues, because if you want to sequence a strain, you have to get that strain from that location. And you imagine if that laboratory is sitting in the US and I have to send strains from China, that's very difficult to export strains from China. Sometimes you'll have to just send the DNA and not the strain. So you run into, that's the downside of having it in house, but just have it in one location. Whereas if you're local, you can say, I've got an academic partner in my country, and it's easy to do that. So I'm maybe not giving the answer, but I'm trying to say sort of like it's horses for courses a little bit, depending on what you have. Nestle is big, and we can do it, and we get enough economic benefit from doing it, but we only still put it in one place.
Okay.
I think I'll add slightly to that, if I may. Because John is talking about a large organization. I think for a smaller organization, inevitably you've got to rely on the laboratory or the research partner for the bulk of the technical expertise. But I think it's so important then that somebody within the company is trained enough to understand the results that are being shared with them. And I think don't underestimate the new breed of master's food safety student who they're using these tools in their studies. So people like that will have a very good grasp of the technology, and certainly what the outputs and how they should be interpreted.
Okay, thank you for that. Purnika, did you want to add anything, or?
I can just add a little bit related to the data analytics side, not the sequencing side, because you would need good computational power to do that. So that's another challenge, is like a lot of institutes and organizations have, and the skill level, like Nick mentioned. So PhD students, master students, they are great, because they are quite encouraging to learn this new technique, how to do data analytics. And at the same time, you must have experienced bioinformatician or data scientist to guide them, or the supervisors can do that. So when it's come to private companies, so you need to hire a data scientist or a bioinformatician to do that, which is quite costly. So those are the key challenges I would think of when it's come to data analytics.
Okay, perfect. Thank you. Thanks for that, Purnika. Okay, there's another question here. If the panel are okay to go on for a few minutes, now we're slightly over time, but if you're still free for us to try and get through the rest of these questions, will AI and machine learning increase the value of genomic data?
I will jump in. Absolutely.
Yes.
I mean, the most forward regulatory authorities are now using tools like that to scrape the databases of isolates every time they put a new one in, looking for connections, looking for patterns. And also the opportunity, I suppose, building on what John said about looking for virulence markers or genetic indicators. There are tools now being developed by researchers to predict the possible behavior of an organism just based on its genetic content. So, a lot of those are artificial intelligence-based tools that are being developed. So it's very much going to build on the data that's being developed through the sequencing program.
Okay, Thank
I
you.
agree that, in AI, the application of AI in food safety relies on good data, and the sequence data is probably as clean as you can get. It's because it's the full sequence of a one particular organism. So the data is pretty robust. So it's, you might say, ideal for the application of something like AI to make, as Nick has said, these connections, trends, and make these predictions.
Okay, great. Thank you for that. And this next question, I think it's already really been answered in the discussion and in some of the presentation about using it to look at corrective action. So in the interest of time, I will skip on. And what should be considered when sharing sequence data with public databases like NCBI or ENA?
I think it's related to what type of data sharing agreement or data governance framework you have in your institute or organization. So public databases like NCBI, EBI, they can hold your FASTQ, like the DNA sequence data, so which you can embed it with your metadata. For example, if you take a FASTQ or the DNA sequence from a clinical sample, you can include where it's been isolated, the location information, but you don't really need to give the patient ID or very sensitive data. So there is a kind of a filtration data that you can deposit in the public database. So it can be quite straightforward if you have clean, good raw data, but you need to be very careful with what type of data you are disclosing to the public databases.
Thanks.
I think from industry perspective, it's a very sensitive topic. I could answer it with 3 words that speak to your lawyer. So generally this is a sensitive topic, for industry to share sequence because there's litigious, angles to it as well. Purnika talked about sort of some of the metadata, in terms of if it's patient or that's a different level of data sharing. But the industry, you end up who will use this, and it's in a public database, and you have metadata that relates to a plant, relates to a product, relates to the sample type, etc. And someone could actually use this in the incorrect way and publish something. So there's those elements, and then there's this sort of timely relationship between outbreaks, and this has happened in the US, where the US have been able to trace back 10 years from the same plant, and a number of sporadic outbreaks. Now, if that plant's openly sharing, they were not openly sharing. This was based on swabs done by the authorities, so it gets into a bit of a sensitive topic when it comes to private sector sharing of sequence data. Legal teams generally have a big say in this.
Okay, excellent. Thanks for that insight, John. Now I have another question that has a couple of parts to it, so I'll start with the first one. There's a comment on the level of knowledge and tools and detail available and accessible to decision makers. Can we expect more accurate risk assessments in the future and less food wasted?
I'll comment initially on that. If you'd asked that 10 years ago, I would have thought it wasn't going to get anywhere. But it is interesting, as I explained, that the FAO and the WHO within the last 2 or 3 years have begun to recognize the difference between different Listeria monocytogenes clones and the potential risk that they pose from a public health point of view. And for that very reason, they're proposing a model, particularly for developing countries, where finding a relatively benign clone at less than 10 may not trigger a recall, whereas finding one of the hypervirulent clones, regardless of the level of enumeration, would require a recall. So I actually think the thinking is moving on, maybe too slowly from an industry point of view, but there is that understanding, particularly in the FAO and WHO, that the risk assessment possibilities are real to manage withdrawals and to manage food waste.
I agree, because I think definitely WGS is going to provide better risk assessments, that's for sure. And you've seen, as Nick has said, with Listeria, you've seen the same with Aztec E. coli. You've GEMRA expert reports who have detailed, depending on the virulence factors that an isolate may contain, they pose different risks, from anything from fatality down to mild diarrhea, etc. So, however, the issue is the legislation or the regulations not keeping up with that. You will still find the legislation will say absence of Aztec or absence in 25 grams of Listeria monocytogenes. So while we're getting more in-depth information from WGS and other similar tools, as part of risk assessment, the legislation has not that flexibility in it as yet. And it's a difficult thing. I have this discussion with a lot of my peers at times. Am I going to convince someone that we have Listeria in our product, monocytogenes, but don't worry, it's not one of the worst ones? That's a difficult communication topic for industry. If the legislation is telling me you must have absence of Listeria monocytogenes in 25 grams, generic monocytogenes. So that's a little bit, whereas the tools are giving us definitely more information on risk assessment. I think the legislation is probably not keeping up with that.
Okay. Great, thank you. And the next part of this question is relating to how risk assessment can be applied to traditional fermentation processes, where we understand that routine sampling using WGS will show up virulence factors which further on in the fermentation process will be negated. And is there more research required in this whole area of fermented foods? I don't know who wants to take that one.
But the whole idea of finding virulence genes, because we have the tools now to find them, but what impact they may have is an entirely different question, and what does it actually mean? So in fermentation processes, you can get lots of organisms growing which might have virulence genes, but what does it mean actually from a public health perspective? So I think it's a difficult one. It's a bit like what we talked about earlier, the virulence genes of Listeria monocytogenes. Some don't have a certain gene, so they're less pathogenic, let's say. It is a little bit the same with finding genes in a whole soup of bacteria and genes. What does it actually mean, even though they may disappear? Are you chasing molecules or are you chasing the organism? That's a little bit the question.
I would agree with John. It comes back to the isolate. Is the organism actually present, or are you finding fragments of DNA as the bacteria are being opened up?
Yeah, and there's another question here which I think we've probably addressed already, but just again, do the speakers envisage a time when the legislation will have criteria for absence of Listeria monocytogenes strains with specific virulence genes? So do you think that we will reach a time where it's focused on specific virulence genes rather than just all mono?
Look, I hope it will, and there is a bit of practical sense, as I've spoken about, around that in certain territories. But look, the research is still very much up and down in terms of being very clear on what virulence factors lead to a particular behavior, or whether there's combinations of those virulence factors. So I think it's still a dream that's pursued by researchers, but but we're some way off that yet.
Okay. Thank you. Okay, another question is the pathogen, does it have to be isolated from these sample before they go on to the next steps, like DNA extraction, or can it be performed from the food sample directly?
I would say that you need an isolate first. Now, there are techniques where you can go straight into the food sample. Now, that's sort of an extension of the metagenomics and the microbiome work that John speaks about. But the validated pipeline certainly would start with an isolate. The labs really do want to work with an isolate.
Okay.
Yeah.
great.
Because even in some, and again, I talk from a global perspective, we have even where you do PCR testing, for example, and you have a positive, and it's a PCR method that's totally validated, by the ISO standard against the classical method. And with some authorities, we report to authority, oh, we've found a whatever pathogen in a sample, and they say, show me the isolate. And we say, no, it's a PCR positive. No, but show me the isolate. So even in some countries, we still, isolation is king. Despite even PCR has been around for 30 years and validated methods, because there's many reasons why you can't get an isolate and you're a PCR positive. So that's why when you go this direction, again, you see some people will still think isolate is king despite these other methods.
Absolutely. Okay, there's another question here on has the confidence in the use of WGS by industry reached a stage that industry is happy to share their data with regulators routinely? We've probably kind of discussed that.
Yeah, it's a little bit. I can tell you, it's always a sensitive topic. This is always a sensitive topic. From our perspective, I can tell you, we have shared sequences with various authorities when asked to do so.
Okay.
So that's about as far as I think I can go. But
yeah. Okay, I understand. Okay, we'll just move on. Purnika, there's a question for you on some of the on the work that you were presenting. Have you analysed the data at a food category level, and if yes, what did it show?
I think food category means like whether it's break down into meat or fish or dairy, or I assume so.
I see.
We actually analyze part subset of our Northern Irish isolates, looking at the ready-to-eat meat product, raw fish, or like whether it's coming from a swab in a food processing environment. So only very few isolates been analyzed in that way, because most of the isolates or the DNA sequences that we got from public database, we didn't get a handle of that type of data. But I think I don't have like all the answers right now, but we did analyze part of our data, break it down to the food types. Yeah.
Great, thank you. And here somebody's making a comment to say that last year the use of WGS in a monitoring program for a small food producer with no epidemiological context led to the destruction of over €500,000 worth of food ordered by the regulatory authorities. So again, making the point that the appropriate risk assessment is key to the application of the technology. Otherwise, it becomes a more a question of potential waste of food. Well, we were just talking about it, hopefully reducing waste of food, but I suppose the potential is the other way.
And I think you still need epidemiology as one of your legs of the stool. WGS is a forensic tool, but you still need some form of epidemiological link as well.
Yeah, absolutely. And another question here from somebody. I've found some really interesting insights in data when sites do WGS on Listeria monocytogenes and Listeria species, usually innocua. You can glean some indicators as to routes through the site. May seem overkill, but would be interested in your opinions on that. I think that's slightly been touched on, but I don't know if Nick, you want to say.
Well, I think that's, I think that echoes the experience that those of us who use the technology have, that it is, you over time will find connections, will learn routes, and it tells you, it informs your facility design and your hygienic design, as well as your ingredient supply chain patterns. So yeah, that's very much echoing the experience of those who will be using it routinely.
Absolutely, thank you. And how long can an isolate be stored by the laboratory before requesting WGS?
The isolate's pretty intact, so it's depending how you store it. People put them on beads, people store them on plates. One example which we had where we had a p-test sample, a precipitancy test sample, was performed in January. It was kept on plates because the laboratory wanted to see the end result when the p-test results came out. Meanwhile, 6 months later, they were testing some product and found a positive. and we discovered that the plates that had been sitting around for 6 months in the laboratory from the p-test were being thrown out at the same time as our samples were being tested. So we picked colonies of the plates, which are 6 months old p-test samples, and they matched exactly what was supposedly found in our product. Another example of laboratory cross-contamination, but in the laboratory was defined, said this could not have happened, but the proof was it was a rare serotype to begin with. I said this is unusual to be found in a plant, and then when we looked at the plates that were there from 6 months earlier, we are still able to get out the DNA, intact.
Yeah, I'll give a slightly different answer. I think it's important to understand the protocols of your laboratory that you deal with, because they may just hold the isolate for a very short period of time before they destroy it for obvious reasons of just keeping those things moving. So if you get into this, it is important to understand how long they're going to keep them for before you need to get them somewhere else for safekeeping, or come to an agreement with the laboratory to bank them for a period of time. But just as an anecdote, I would echo what John says about the unbelievable life of isolates, because we've had some get lost in the post and turn up 3 months later, and we still manage to recover DNA. So they should be stored at -80 or whatever it is, but they can still survive.
Excellent, thank you. And I think we're on the last question, and somebody is just asking John about if it's possible to get a link to the Listeria court case you mentioned. So if you want to send it on to us, we can maybe send it we can send it out. We'll be circulating the
It was in Estonia. You can probably Google it. It was in Estonia. I think it was tuna or salmon fish or trout maybe producer, of fish. So if they go, I'll send it to you anyway. But
yes.
if they wanted to urgently, I think do Estonia producer or packer Listeria.
Lovely, thank you. And I think we are finished questions, and sorry we went over time, and thank you so much to the panel, both for your presentations and for your patience and thoroughness in answering all of those questions. That was a really, really informative session, and I think people will have learned a lot from it, and a great discussion as well. And thank you to everybody who sent in questions and participated in the conversation insofar as you can through a webinar. So I will just